Okay, hi guys. This is the second lesson in this little GCSE series that I'm putting together while we're all off school. We're gonna plow on with cell biology. So yesterday we had a look at cell organelles. We had a look through prokaryotic cells and eukaryotic cells. And if you didn't get a chance, they're all on the YouTube channel that you should kind of hopefully be watching these on. So I'll kind of log each one and you can go back and do it. At the end of this lesson, there's going to be some follow-up questions, and I'll show you where you can get those, and you can find the mark schemes to kind of self-assess and see if there's any issues with them. Again, if you kind of put any questions in the comments section, I'll try and get round to them and answer them when I can. So today we're going to be having a look at cell specialisation and differentiation on the AQA specification. And then we're going to be moving on to culture and microorganisms. Now, if you're doing AQA, the culture and microorganisms is just for triple scientists or separate science biologists. So if you're a combined scientist, what I'd suggest you do is just maybe fast forward to the end of the video and find out where the questions are, and then you can just kind of plow on with those and have a go at those. So remember, I'm following the AQA specification here. So if you are at Excel or you are possibly OCR, very few schools up here do that. But if you are, you could maybe just kind of correspond the broad strokes of what I'm giving you here, the kind of the knowledge, and apply it to the specification. But I'd be referring to this document. Most schools up here in the Northeast are doing this specification though, so this should hopefully be relevant for most people. So if you have a look at these spec points, this is what I'm going to be covering. So maybe you could have that document up in front of you, or maybe even if you're really organised, you've got it printed out and you can sort of tick things off as you're having a look. So we're going to talk about or build on what we did yesterday and talk about cell specialisation. So if something's specialised, what that means is essentially it's specialised to do a particular job. So if we take a sort of classic example of a specialised cell, which is a sperm cell has a few sort of unique features that make it adapted to do its job. It has a tail, and that allows it, or a flagella, that allows it to swim towards the egg cell that it's going to fertilise. It also has, at the head of its structure, something called an acrosome. And an acrosome is basically filled with digestive enzymes. And those digestive enzymes will be released, and they will break down the outer layer of the egg and allow the genetic information that's inside the sperm cell to enter and fuse and cause fertilization to happen. It also has a collection of mitochondria. That's not a very good mitochondria there. And the mitochondria are providing or releasing energy from aerobic respiration that's allowing the tail to beat or to move. It also has this kind of streamlined shape that allows it to move faster. So all of these are specialisms of that particular cell. We might also think of another really common example in example questions of an example in plants. This is a root hair cell. A root hair cell has a couple of adaptations that we need to maybe be aware of. The first being that it has this large surface area. Very, very common answer, very common idea in GCSE biology. If something has a large surface area, that enables it to carry out more exchange. Let's imagine that the root hair cell here looks like this. You can see there, this has this huge appendage here that enables it to increase how much exchange is going on, how much surface is there available for things to move in and out. We've got less of a surface area if the cell looked like this. So a root hair cell is something that's found in the roots, and that cell is responsible for the exchange that takes place in the roots. Now we've got two types of exchange. We've got osmosis, the movement of water, which is something we'll come across in another video. And that moves into the roots and up the plant to all the different parts of the plant that will use the water. And then we also have active transport taking place. Now you will have diffusion also. Again, we'll cover transport in more detail in another video. But we get active transport of minerals into the roots. So they move into the plant through these root hair cells. So often we'll talk about cells that are carrying out active transport, and this will be kind of relevant to the question. This is the main adaptation that often comes up in questions about root hair cells. But we'll also see whenever we have active transport, which requires aerobic respiration, we also have mitochondria to release the energy needed for that active transport. Again, we'll talk more about that when we do a video on transport. So root hair cells have these specialisms that enable them to do their job, to absorb things from the soil and move them into the plant. So we have a few other examples on the spec there, a few other bits. There are other plant cells that we need to know about, xylem and phloem. Xylem are tubes that move the water up from the, up and down the plant. They are hollow tubes, they don't have any organelles, and that means that the water is not impeded as it moves up and down. So xylem in a plant. We also have tubes called phloem that move sugar up and down the plant. I say up and down because really the sugar is made in the leaves and it moves down when photosynthesis takes place. But in situations where the leaves have been removed, in young plants or in plants that have lost the leaves, etc., then the sugar is being released and moved up the plant from other stores, like in the roots. So the phloem are responsible for moving sugar, and they have separate cells, companion cells, that contain organelles for active transport and movement of those cells. But again, they don't have any organelles in the middle, and that stops the impedance of any kind of flow of the sugar up and down the plant. We also have a couple of other examples of our specification here. I'm just reading closely as I'm doing this. We've got muscle cells and nerve cells. Now, nerve cells are to do with communication, and this is the sort of classic example of one. There's a few different types. A nerve cell carries an electrical impulse from one part of the body to another, the nervous system. So it has these dendrites, these extensions that come out, and that increases the surface area, allowing other dendrites that are connected to other nerve cells to make these connections. So these are the dendrites here, these extensions that make connections, the nerves. They also have cell bodies that contain a nucleus. These have extensions that are coming off to make connections. They have this long, thin body in the center called an axon. We do know more about nervous transmission later on. So they have a very large surface area to enable them to make these connections. We also have muscle cells, and muscle cells are involved in contraction. The ones that are in the skeleton, skeletal muscles, are involved in movement. So muscle cells will have lots of mitochondria, and those mitochondria are carrying out aerobic respiration to release energy and enable muscle contraction to take place. So there's a few sort of one or two adaptations that we need to be aware of each of these types of cells. They're all specialized to do particular jobs, but how do they become specialized? Well, they differentiate. Now, all differentiate means is they change from being kind of a template, like a moldable, changeable kind of blueprint, if you like, to becoming one of these specialized cells. All of our cells start off as stem cells. So if we imagine a sperm meeting an egg, they fuse together to form what's known as a zygote. And a zygote contains the genetic information from one sperm and one egg. Now, that zygote will then divide, and it's something called a blastocyst, which will eventually form an embryo. So this embryo is the beginning of a baby. Now, in the embryo, this cell here might become the leg, and this cell here might become the brain or the heart. So all those cells need to specialize to do different jobs in exactly the same way as we've been talking about here. They might have different shapes, different structures, but really importantly, they're going to have different kind of organelles that support whatever job they're doing. So these embryonic stem cells all have the potential to become any type of cell in the body, and they have to be kind of directed and changed into certain types. Now, once they have become specialized, once they start to change, they can't go back to being other types of cell. So cells that are in the heart can't then change to become cells that are in nerves. And this might be a really interesting thing if you've got a bit of time on your hands to read about. Stem cell therapy and stem cell technology is something in the future that might kind of shape the way medicine looks and really help us change people, because we think that stem cells, they have the potential to become any type of cell, might help us treat people who have brain damage or people who are paraplegic, who damage the spinal cord, because we can replace tissue with these kind of templates. So a stem cell, we're putting it in a glossary or a definition, is an undifferentiated, an unspecialized cell, and it has the potential to specialize into any type. Now, when they differentiate, they change, and the organelles and the structures within them become adapted. Now, there are two types of stem cells in animals. We've been talking there about embryonic, but we have other types of stem cells that exist in our bodies as we go past this 8-cell stage. After this 8-cell stage, stem cells aren't fully undifferentiated. They can become certain types, but not every type of cell within the body. So we keep a certain amount of templates within the bone marrow, for instance, or under the skin. They're called adult stem cells. And what these adult stem cells do is they can change into a few different types. So, for instance, the bone marrow makes blood cells, and it can make a few different types of blood cells, and they can specialize and change to make these different types of red blood cells depending on what the job is. For example, a white blood cell and a red blood cell do two different jobs. The bone marrow can kind of specialize to change these different types. So we need the ability to make some sort of types of cells that we're going to use again and again. We use lots of blood cells, and we need the ability to make a few types, but not every single type of cell. For example, cells in the bone marrow can't make nerve cells. They can't make muscle cells. They can only make a few different types of cells. But embryonic stem cells, as we've mentioned here, have the potential to make absolutely any type of cell. Now, plants, it's worth noting at this point, have their own type of completely undifferentiated cell. So a bit like our embryonic stem cells, these are called meristem cells. And you'll do more about this when we do cloning. Plants can very easily be cloned because they have completely undifferentiated stem cells that can be changed into any type. So, meristems are plant stem cells. We get them generally, and they're all over the cell, but for our intents and purposes, you see them in the roots and the shoots at the tips where we would find that meristemic tissue. So this is cell specialization, cell differentiation. If you're one of those people who is not a triple biologist, I suggest you kind of fast forward the end of the video and find out where the questions are, have a go at them now. But if you are one of those people, we're going to move on now. On the AQA specification, it's 4.1.1.6, culturing microorganisms. So this is one of the required practicals that you have to carry out. And it's essentially looking at the effects of antibiotics on microorganisms. Now, microorganisms grow by a process called binary fission, which isn't the same as mitosis if you're reviewing this video, having already done a bit of GCSE content. Binary fission is where it's a form of asexual reproduction where one cell will split into two identical cells. And in bacteria, usually that takes 20 minutes is sort of the gold standard amount that we use and we teach. It's different for different bacteria, but it takes roughly about 20 minutes, which means it's very, very fast. So that will only kind of take place, that binary fission, that split, if the bacteria have enough nutrients and the right conditions, for example, a suitable temperature, enough kind of nutrients within the growth medium that they're growing on to enable that growth to continue. Otherwise, it might take a little bit longer or happen a little bit faster. So we can grow bacteria in two sort of mediums. We can grow them in what's called a broth culture, a nutrient solution, if we're going to have lots and lots of it. So that's basically kind of like a flask. You might have seen this in lessons, you might have done it. If you were kind of plating it out, you'll have this kind of, looks like kind of chicken stock or something in this flask, and you kind of plate them out onto agar plates. So an agar plate, or agar, is a growth medium used with gelatin or seaweed components. So agar is just a growth medium, and we pour that agar onto a Petri dish, which is that kind of plastic circular dish with the little lid on it, and then we tape it up. We'll talk more about that in a second. But what we do is we pour the agar onto the plate, let it set, and then put the lid on it to make sure there's no contaminants. We'll talk more about that 1 minute. And then if you've got a nutrient broth like this, you put the bacteria onto the nutrient agar, so they have the nutrients they need. We grow them, and then we see colonies of bacteria appear on the plate, and we can look to see what kind of different effects like variables might have on the effect of how well they grow or how they react to different kind of substances like antibiotics, things like that. So we place them onto the plate. This colony that we're looking at here is essentially kind of millions and millions of bacteria. 'Cause bacteria are really small, right? So we're looking at lots of those bacteria all kind of collected in one place. So the whole kind of point of this process here, these are kind of two ways that we've got of growing the bacteria. We can grow them in colonies on plates, so here, or we can grow them if we're going to have like kind of lots of it in a solution, like a liquid solution in a broth culture. So what we've got to do is, if we're having a look at the technique, the practical, we've got to be able to use something called aseptic technique in order to grow the correct bacteria and not get any contaminants. And contaminants just basically means you've got other microorganisms. This might not necessarily be bacteria. Quite often it's mold or fungus, or it might be like other types of microorganisms that are in the air on surfaces. So what we do is we take a plate and we kind of sterilize the plate before being using it. And sterilize just means kill any microorganisms. That can sometimes be done with steam, or flaming things, putting things in a loop, or using like Virkon or disinfectant to wipe surfaces down. And that just gets rid of any kind of potential contaminants that might get into the sample. So then we're going to have Bunsen burner. We're going to take what's called an inoculating loop, which is just basically a metal loop. We flame the outside of the loop until it glows red, and we know that all of the bacteria then have been gotten rid of. And we also sometimes see in questions talk about the Bunsen burner kind of being left on. And what that does is it causes the air to circulate around the area. So if this is my bench and here's my plate and I've got a Bunsen burner going, then what will happen here is that the air, because of convection currents, will kind of rise and fall, become more dense and less dense. And any bacteria that's in the air will kind of circulate and never sort of land on the plate. So by having the Bunsen burner on and leaving it on while you're doing it, you're also kind of stopping any potential contaminants. So we flamed our loop, we've kept the Bunsen burner on, so we know the loop is clean. What we then do is we place the loop into the culture. We take a swab of that and then we streak across our plate, and then we rotate the plate kind of 90 degrees, and then we streak again and we rotate again to give us another rotation, and again, and that should give us a nice even coverage all over the plate. And what we're making there is a bacterial lawn. Now that will give us a lots of colonies hopefully that will appear on the plate, and then we can test to see how that works. Now I'm talking there about using an inoculating loop, a metal loop, but you can, and quite often schools will use just cotton buds, sterile cotton buds, It's the same principle, you just obviously wouldn't flame the cotton bud because it would be on fire when you were doing it. But if you do that, then it will give you exactly the same kind of process, same results. Once you've got that, you place the lid onto the plate. We've been very careful when we were plating that to lift the lid just a little bit to allow the loop to fit onto the agar plate, and that stops any bacteria or microorganisms that are in the air falling on the plate. So quite often when I'm teaching this, students will forget and they'll have the plate kind of just off the plate, and I'll say you'll have to start again because all the bacteria that's in the air is falling on the plate. So you lift that up just a little bit and then place it loosely. And then what you do is you place sellotape, tape up the plate at the edges or in the kind of four corners. And what that does is it allows the plate to let air in, because the bacteria need oxygen for respiration, but it's going to stop the plate from lifting off or being lost, and that's again going to kind of prevent any contamination at any point. We're then going to put it into something called an autoclave, which is going to keep the temperature just above room temperature, and that's going to give the right conditions for the bacteria to grow. It's going to provide optimum conditions. Now we don't want it exactly at body temperature, sorry, not room temperature, kind of more like 40 degrees. We don't want the temperature to be like body temperature. Body temperature is bad, because if we kept it at body temperature, that would mean that microorganisms that grow and survive, i.e. things that could do us harm, students harm, would be growing on the plate. So we do it slightly higher than that to make sure none of those kind of pathogens would be growing. We're doing it in a school lab setting. So we've done all that. That's the aseptic technique. They're all the bits and bobs that we've incubated at. We'd essentially, so we've talked about how they'd be cultured, incubated around 25 degrees, and our body temperature wouldn't be so bad, wouldn't be so good. And now we're going to have a look at the results and what might be for that. So I'm just scanning down the specification here. We're going to have a look at the antibiotics and the areas around the plates. So let's imagine this is our plate. What we might have done there is use a China graph pencil to draw a section on, and then once we've got the plate and we've incubated it, before we incubated it at 25 degrees, what we're going to do is slip on a disc of paper that's been soaked in an antibiotic and place it in different sections of the plate. So what this might be is that these are different antibiotics. So this might be penicillin, and potentially this might be something like methicillin, something else. And we place these different antibiotics, and what we're doing is we're trying to see what the size of the clear zone is around the outside of the antibiotic disc. So what that means is, if there's a clear zone around the outside, that means no bacteria have been able to grow. So the antibiotic has killed those bacteria, and we can see how effective the antibiotic is based on the size of the clear zone. So different antibiotics have different efficacies, or effective against different types of bacteria, depending on the type and the biology of each microorganism. So they will sort of kill them and get rid of them in different ways. Now, we can measure that using the cross-sectional area of the circle. So if we do pi r squared, that will give us the area of the circle. And even if the circle kind of in question, sometimes the circle might not, strictly speaking, be all the way, exact. What you can do is you can use just the radius and sort of make it a rough estimate of what the circle would be, using that same formula. If you have like an incomplete circle, or you have an incomplete clear zone that's around the outside, you would use the same equation to make a same rough judgment. Now, sometimes in questions, this practical might not be different types of antibiotics. It might be different concentrations, which would represent how many days you were taking the antibiotic. So if you had different concentrations, this might be 25% or 0.25. This might be 50%, 75%, 100%. So what that might represent is the number of days that you've been taking it, or the length of the course of antibiotics. And it would be the same principle, the same experiment, Because when we test for antibiotics, we not only need to know which type, but we need to know how long you've got to take the antibiotic for it to be effective. If we take antibiotics for 5 days and we reach the point at 2 days where they've had their maximum effect, there's no point in taking them any longer. So you'll do more about antibiotic resistance later on in the course. We'll see that in another video. But for now, this is all we need to know about the key practical and culturing organisms. So I'm going to show you where you can find the questions on this practical. It's worth watching like a little YouTube video to show, to just illuminate some of the bits of equipment that I've been talking about there, like the loop and the flask and the agar plate, and setting them up. There's lots on YouTube that you could find. If I were looking for some questions on that here, I'd then be going on to the website I showed you in my video yesterday, which was this cell biology section of physics and maths tutor. Really great website, lots of good resources. And if I have a scan through the cell structure section, I'm going to see some questions there around, just in case I kind of mapping around and scrolling through some cells on specialisation within those parts. Here's a good one here on antibiotic resistance that we've just been talking about. So you might have tried a couple yesterday if you went on and had a look at the cell structure questions and then marked them. It might be that there were a few that you couldn't do or a few that you remembered, just in case of having a look at that. Okay, tomorrow we're going to be doing cell division, mitosis and the cell cycle, and I'll upload this video now. Obviously you don't have to be doing them when I'm recording them, you can just watch them whenever. Alright, thanks, see you tomorrow.